Bradley.
Hi, how are you?
One of my not well. One of my sons had a seizure last night.
Oh, wow. Sorry.
Okay, and that's why I'm late. I have to take a shower now,
Okay.
so I'll be a little bit late.
All right.
Okay, I'll be in about 15 minutes.
All right, thanks.
Okay, bye.
Bye.
Yeah.
Well, hello.
Okay.
Yeah, hello. Welcome to the meeting. Yeah, sorry to hear that. So, I guess we're waiting for other people to show up. I'm sure we'll have a few more people.
Yeah.
Is there anything anyone wanted to say? Anything they wanted to present, or?
I'm trying to pronounce Mr. Lee's name. I think Jiahang is. Have I got that right? Or anyway, hi. And I'm just. This is a really nice camera I've got, and I did put the microscope together. I just have to calibrate it. It's frustrating. So, do you want to see it?
Yeah.
It's right over there.
Yeah.
Yeah.
So this is the camera one of the cameras Susan's been working on for. Yeah, that's a good view right there. Yeah.
So it's just there, and you have to put your peppercorn or whatever on here, and this may be too tall, but I put it there and then line up the microscopes, and I've got this time I have 4 screws so that I can line it up, and they're on a spring, so you can change the angle of these. So this is the new one. It should be easier to work with, but so far, anyway, yeah. So this is the bottom, and the top's just over there. I put it together at least.
Yeah. Well, that looks great. Yeah. Nice to see that coming together.
Yeah, I can say that I did something this weekend.
Yeah.
Yeah. Usually I don't get as much done as I want.
Well, that's good. So I guess we'll get started. Yeah. Is there anything else anyone wanted to present or talk about? I know we had some things that we had mentioned in the last several meetings. Otherwise, I've been talking to some of you about the GSoC projects. I know I've been talking about Jiahang about the CL or sort of the GNN's approach, and he has an approach that he'd like to write up. So I'm going to look over your proposal later on today. I'll give you some feedback, and then the submission actually is tomorrow for this. So people, I think, are kind of hitting the deadline, but that's okay. We talked about it, and if I know that like a lot of people are planning to submit. So if you don't get selected for it, I'm still open to working with you, over the summer, and working on a project and getting something really nice as a product of that. Now, that being said, we have a lot of data sets that you can use. And so this is an opportunity for people to use their skills and build a project out of it. And then if you get selected for GSoC, all the better. I've talked to Karan as well. He sent me something. I'll review that. Hare Krishna, I've reviewed his things. So that's pretty good. We have a number of people also, I think, who are maybe submitting things, but I haven't really seen a draft from them. So we'll see what happens from that. Yeah. So let me. You had something to say, Susan?
Sorry. So the deadline is tomorrow.
Yeah, for GSoC. Yes. So the proposal.
Okay. Deadline. Well,
it's not your. Yeah.
I have another deadline. Anyway.
Yeah,
I'll
that's
turn
not for
off
you.
my camera and
Well,
stop.
I know, but not another deadline. No. So yeah, yeah. So I'm gonna actually talk a little bit about some of the data that we have available. So I've talked about we talk about C. elegans, we talk about other organisms. C. elegans is a nice model. It's probably the model we've worked with most in the group. We have a lot of data from C. elegans available. Like we have cell tracking data, which I think Jiahang really is interested in working with. And that's very common actually in a number of organisms. So we have cell tracking data where they label the nuclei with some fluorescent marker. And then they take microscopy images of the organism, usually the embryo, and all the cells are the nuclei have some marker in them, and they can track those nuclei under a UV microscope, and they can follow that through time. So they just take images, so many images per second, and then they have these focal plane slices, which means that they go through the specimen and they go through different focal planes, and they get those images. So you end up with a stack of images through the embryo at different sort of levels, so you can see different, if you go from like the top to the bottom, so you're going, from the dorsal to the ventral in anatomical terms, and you're getting all these slices and you're getting images from those different places. So sometimes cells are towards the top, sometimes they're towards the bottom. You can figure out, you can get all the information about the cells that way. But you also have a time series. So you have, a number of frames acquired per second, and then you just simply stitch those together as like a time. You can treat it as a time series. You can also just look at like different cells when they emerge and look at their position and do it that way. So you don't need to have a time series, but you can also do like a dynamic analysis of it. Now, there are a number of data sets that exist where they don't have the images, but they've extracted the data from the images, and they've created like a data set, a raw data set of points, and the points are labeled by coordinates. So when they take an image, their coordinates, these points are different coordinates on the image, and then they give you this list of coordinate positions, X, Y, Z, which would be along the three anatomical axes. So it's anterior, posterior, left to right, laterally, and then dorsal to ventral, top to bottom. So top to bottom would be Z. X would be the anterior, posterior, front to back, and then the sides would be Y. And so that's how they usually do this, and that's how the data is presented. So you can get it out of the raw data, or you can get it out of the numeric data. And then what we've done in the past in the group is to transform that into some sort of coordinate system of that you can use to compare across different images. So the raw data just basically has some coordinates, some coordinate labels. They don't really make much sense if you compare them across images. But if you use a sort of an averaging for the sort of like a centroid for the entire image or something like that, you can usually get a nice coordinate system that's consistent. There are other ways to do it as well, but that's what you need to do. Those are the steps you need to do to get some of the cell tracking data in. So we've done a lot for C. elegans. There's some for Drosophila. There are different model organisms that people use. We can get cell tracking data for that, for zebrafish. And then we've also worked with other organisms such as diatoms. So we have data from diatoms, which are these single cell algae. Sometimes they live in colonies, and we've been analyzing the colonies. There are other, organisms that we've used, like, more exotic ones that aren't really usually model organisms. They're very good images of spiders, spider embryos. There are also some good images of plant embryos. And so we don't have any of those in our We haven't worked with those too much, but those exist. Hello, Karan. How are you? So let me go over some of the...
Hey, hi. Hi, Bradley.
Hi.
Hi, Susan. Hi. Hi, Jiahang.
So let me go over some of the data that we have, or some of the things that we like to use for data sets. So the first one is this systems biology database. System Science of Biological Dynamics database, to be specific. So this is run out of Riken in Japan. And this actually... Ken Ho, who used to attend our meetings from time to time, he was running this database, and I don't know if he's still involved with it, but it's run out of Riken in Japan. They have a lot of different microscopy images, and they have this this XML protocol which they use to sort of extract the metadata and store it. So they have a nice set of data, a nice group of data sets here. If you want to learn more, you can read this paper, SSBD, a data set of quantitative data of spatiotemporal dynamics of biological phenomena. And that paper is about everything I just told you about, like cell tracking and things like that. Because a lot of these data sets are cell tracking data sets, more or less. They have different things, like you can look at very specific structures here, like the oocytes in C. elegans. You can look at nuclear division in an entire embryo. You can look at retinal tissue in human embryonic stem cell cultures. I think this is either cultures or somewhere in vivo in an embryo, but I don't know, or in a developing human. This is Donnie O'Ririo. So this is nuclear division dynamics. This is the zebrafish. This is the nuclear division dynamics in C. elegans wild-type embryo. So this is another type of image that you can get. So there, we look at some of the images that we've seen in the group. We have this sort of the whole embryo data where you're looking at cells dividing. And this doesn't really label the nucleus, but the nucleus would be somewhere inside the cell body. And, if you're looking, this is a DIC image, so you don't see fluorescence in it. But you can see fluorescence in some of these other images. This is calcium imaging protocol. And then this is another type of imaging. This is molecular dynamics imaging. So there's a lot of potential information you can get out of microscopy images. If you're working on a method for machine learning or a method for, graph neural networks, you can actually use your method to be a much broader set of things. When we were working on DevoWorm, for example, our goal was pretty specific. We wanted to create a pre-trained model for something like this, where we could take a whole embryo and, sort of look at the cell nuclei and, we treat them as sort of centroids in a space. And so those centroids would increase with time. They'd move around. And then we could do things like build networks, or we could look at their spatial distribution, or we could, do other things relative to their division dynamics and their cell division dynamics. So, as you had more cells, where are they going? Where are they migrating? Those sorts of things. So you can do all sorts of things from that baseline. And so what we were doing with DevoWorm was we were trying to create a pre-trained model. So you'd have like working from a C. elegans data set, we could just take some image data, plug it in, and then get allow it to segment the cells, first of all, segment the edges of the cells, and then detect that centroid, which was the labeled nucleus. And then we could output the data there. So we could output like the dimensions of the cell and the nucleus. And so that data could be transformed into numeric data. But to do that, usually you need to have a good training set. Well, what we have for DevoWorm is we have, you just plug in these images and you can get the numbers out, and they're pretty accurate. But you don't need to be concerned with like the pre-trained aspect of it. You can actually create a platform that is maybe different than that. Like if you're doing with these graph neural network embeddings, for example, you might take an embryo and you might be interested in doing a different thing with it, encoding some of the network dynamics, for example. And I know that we've talked a little bit about that with some of you on how that might be done. So this is one source of microscopy data. This has the advantage of being, these are published in papers, so some of these data sets are, they may be about 10 years old, but they're, good data sets. They were published in Nature and Science, and, they provide a good, and they're probably newer ones, but the thing is that we want good data sets, so this is a good source for data sets for a number of model organisms, a number of different types of microscopy. Do you have any questions about that, or?
Yeah, I just typed in the chat. Ken Ho has moved to England.
Oh yeah.
And I know Dick and I invited him to come back to DevoWorm, but he wasn't sure about the protocols for that, where he moved to. Too much secrecy, I suppose.
Yeah.
Well,
yeah, I'm glad he's I've seen him once in a while, like in the Slack, but I haven't talked to him recently. But that's good though, that So yeah, I don't know. Well, we'll see if he can get away from the secrecy. But yeah, so this is, and I don't think this has any axolotl data in it. Not really sure. You'd have to search for it. But we've also worked with the axolotl data, and each of these data sets are very different. So if you create a model, you can create a model for one specific type of organism, like axolotl or C. elegans, and that's probably good because that specific organism, it's different enough from other types of organism in their development to be useful, because it's a model organism. But you can also, and I had someone who was trying to work on this and they didn't really get very far, but to have a general model, which is like something that covers a wider range of embryos or biological phenomena. So but of course that requires a lot more data to train. So these are the kinds of issues we have to worry about when we talk about some of these models. Now, the next data set I wanted to show is this. This is our repository on GitHub, and this has some things that are processed. So these are processed data sets. If you go to DevoWorm, github.com, DevoWorm /DevoWorm, and then the second DevoWorm is capitalized, so it's case dependent. You'll see that we have all these folders. So we have different types of data sets. So this is, we worked with what they call a sea squirt, which is an ascidian, and it's a type of organism. It's a marine invertebrate that has a number of different distinct life cycles. And, it goes from like being this fourfold symmetric embryo to being this like tadpole almost, and then it ends up being sessile later in life. It settles down on the bottom of the ocean and it sits there and filters the water for its food. So it has a very diverse life history, but this data is from the early embryonic stage. And the reason that's important is because this is another organism that has a very well characterized early development. So this is like C. elegans. C. elegans has a very distinct early development. You can track every cell, but more importantly, the history of that cell. You know what the lineage tree looks like. So I was talking about this with Jiahang yesterday on Slack, that you have C. elegans has this very distinct type of development where every cell, what the fate of that cell is going to be. It's deterministic. And so because of that, we can track the cells. We know which cell it is. We know what the identity is. and we know what it's going to become. So it's going to divide into two cells. We know what those cells are going to be. And we know it's deterministic across C. elegans with some exceptions for like mutation, that they're all basically going to end up in the same place. So that's an advantage. And actually, the Ascidian actually has the same advantage. And in this README, we have some things. We have this Antecedent database, which is actually something that is used in that community of biologists. This is a database for these Ascidians, and it allows you, there's some genome annotations here. There's some species. So I think they have some data, raw data sets for like the lineage tree, like I think the anatomy tab. But they also, I think there's some, and I don't know if it's in this database, there is some microscopy data. So there's an anatomy tab here actually that kind of goes into some of this anatomy. And as you can see from the anatomy here, it's fourfold symmetry. So when a cell divides, you have these four poles here, and they're symmetrical in that the cells are organized in the same way. So it kind of, they have, like we as humans or C. elegans or axolotl, they have twofold symmetry where you have a right and a left. And then things are, your right arm and your left arm are very similar, not identical. That's twofold symmetry. Now, fourfold symmetry is where if you had an arm coming out of your front and coming out of your back, and they were all identical, that would be fourfold symmetry. Actually, you probably wouldn't have a front and a back anymore. You'd have like four sides. But that, so this is how some organisms, how their development proceeds. And of course, this is different than C. elegans, because C. elegans has this twofold symmetry, which you see early on as this anterior-posterior differentiation, and then you see it more as a left-right differentiation. But this is fourfold symmetry very early in the embryo. And so that's another organism you can look at. So we have some data on that. That's the numeric data for that. We have the C. elegans raw nucleus data, or nuclei data. And this is this is actually from a data set. I don't know why the data set's not labeled on this, but this is one of the data sets that they have on SSBD in terms of the microscopy images. This is just the raw data of this. This is like the numeric raw data. So there's a lot of numeric raw data here, much more than what we have for Ascidians. And this is like, I think this series is from like the single cell all the way out to almost to the hatch stage of, where the worm hatches. It's, the embryo keeps growing in terms of cells, and then there's a hatch where it comes out of the egg, and then there's some postnatal development, which is where they, there's more cell division, but it's not essential to the function of the organism. So, they have this sort of this cell division that happens outside the egg. But this data set gives you everything within the egg. Then there's like cell birth and death timing data for C. elegans. This is where we can take those cell tracking data and annotate them with timing, with differentiation times for each cell. So you have, like, for example, time a cell is observed, a time the cell divides, timing of other things, cell annotations, what the cell is. So, like I said, in C. elegans, every cell has an identity. This cell has a specific identity. It does something. We know what it does. So we have all that information here. We also have more information about the lineage tree and other things. So we have a lot of data on that. We have some other, I think lineage tree database is something else. Well, if you're interested in these data, I would maybe play around in here, but I'd also, I can tell you exactly what those are offline, because I have to see what they are. I mean, I have to, go back and look because I haven't looked at this in a while. But the point being is that I can, we have a lot of data that's been processed. There's a lot of data available, and if you need data, that's not a problem. Finally, there's this DevoZoo, and this is something I've mentioned in the group, and we haven't gone through it enough, but just one more time for people who may not be aware. This is this view on GitHub link. This just takes you back to this. So that's what that is, and but we also have different data sets organized by organisms. So this is for nematodes. This is C. elegans embryo data. This is the lineage tree, cell birth and death timing, differentiation tree, cell position, and the gene expression data set. This isn't exhaustive, but this is the stuff that we've had in the past that we put up here. We also have microscopy movies, so these SPIM images, these SPIM data sets are actually really good for finding, being able to segment cells and finding nuclei. If you're doing some sort of machine learning technique to segment cells, these are very easy to work with, and the nuclei as well. Some of these other data sets are not, are maybe a little bit more challenging, but they have different types of features in them. Then this is for the C-squirt, for the Ascidian. We have microscopy movies, and we have this data set of the cells and their identity, and their, I think, some sort of timing data. I'm not sure if it's by seconds, but it's definitely useful for organizing the lineage tree for that. Then there's Drosophila. We have a couple of data sets for that. We have one for zebrafish. We have one for mouse where you have a zygote to blastocyst data set. It's just like a portion of the development. And then the spider embryo data. So we have some microscopy movies for spider embryos. We have then the Basilaria data set. So these are our diatoms in the data sets that we have for that. We have these colonies that move around, these single cells that are organized into colonies. Then we have even something called morphozoans, which are artificial organisms. And we have some, you can run a simulation of these morphozoans and see what they look like. And then some other things as well. So that's our DevoZoo. And so I'm looking towards improving upon that in the coming months, but I haven't had time in the recent past to do anything with it. I think that there's an opportunity to sort of pick up on this theme of model organisms and the work on model organisms. So I don't know how we're going to improve it. I'll probably talk about it in coming meetings, but I'm thinking that I think there's some use in terms of model organisms. I know there's a lot of interest in sort of comparative studies where people take different model organisms and look at them and look at the differences between them and the similarities. So that there's an opportunity there. And of course, if you're working on something, some sort of software platform, that can be incorporated as well. We have the platforms for the different machine learning data sets, but we have those on a different in a different area of the website. So it's like DevoWorm AI. So we have a library there, but that's something that might be integrated more closely with the model organisms. So, okay. So, any questions at this point?
Oh, hi.
Hi.
Hi, yeah. I have some questions. I'm wondering what the data sets the DevoLearn has worked on. I mean, I have seen its GitHub, and I see that the DevoLearn has worked on some cell tracking challenge data sets. And I mean, if I'm wondering, has the DevoLearn has it worked on the C. elegans data sets before?
Yeah, so DevoLearn has a lot of the development of that was based on SPIM, like the SPIM microscopy, which is it's a type of light microscopy, and we're using specifically a lot of fluorescent images. So the fluorescent images will have it'll have a label on the nucleus, and then it'll also have like labels along the edges of the cell. And so you can actually use those type of images to segment cells and to find the center of the cell. So you have like this tracking of the center of the cell, and you can actually segment the cell out. Now, there are other automated tracking methods that have been used. The data sets that I showed you before about the C. elegans cell tracking data sets, those were actually also used in DevoLearn, and that was used as sort of like a pre-training step. So the idea was that we had, we would take the data from this automated cell tracking and put that in as a pre-training step, so the model would know what that looked like. And those images are, they're good, but they're also pretty raw. They're not very high resolution, and so, but they give you information about where the cells are in the embryo. And then we also use the SPIM data to sort of confirm it at a higher resolution. So we ended up with this, pre-trained model that had a pretty good sense of like how to segment a cell when it saw something in a C. elegans embryo, take out the centroid, and then use that as a center point, and then, give you automated output of the numeric data. So yeah, we used those two data sets. C. elegans BIM data sets. I mean, there's probably more than one. I can't remember exactly which one we used, but those are higher resolution. So we have like these two data sets that we used. Automated cell tracking, and then the SPIM data set, which was segmented by the system. And then if you use DevoLearn, it works best with those sorts of high resolution images. If it's a lower resolution image, it'll still work, but it's not as accurate. So that's how that was the strategy.
Yeah, I understand your point. And in my proposal, I think maybe our project will use the DevoLearn because we need to build the graph structure from the given videos and images, and we need DevoLearn to extract some information from these images, such as the coordinates and such as the centroids, such as the distance between these centroids, and it will help us with building the graph. That's why I asked this question. And I have another question is that actually in my proposal, I designed a project which can construct some high-level embedding of graphs using such as GraphSage and GCN, GAT, some other graph neural network models. But we haven't, because I have little understanding of the biological analysis, so I didn't know what task you will focus on. I was doing some works related to graph neural network. I mean, the algorithm part of GNN will focus on some tasks such as the node classification, graph classification, and some link prediction. And this task will help us to evaluate how the models perform and how our methods perform. But if our project only generates embeddings without using tasks to evaluate the embedding, I think in a way maybe we lack a method to evaluate our project and our methods. Yes, because there are a lot of ways to build graphs, to build node and edge features.
Yeah.
But if we don't have appropriate tasks to evaluate how the model perform, or is the node feature and edge feature good or not, maybe Yeah, that's my point. Yes, I
Yeah.
think maybe tests are important, but I didn't mention that in the project because I didn't think we have enough time to cover the all of the points. Yes, yeah,
So I think that's an important point with in terms of tasks. And you're going to have data, and you're going to be extracting data. And there's actually in C. elegans, for example, there is a lot of annotation. So, like I said, the process is deterministic. So we know what the cells are, we know what the labels are, we know what they're going to become. But it's interesting to just do these tasks independent of that and say, what is this? Because the annotations that they made when they made the lineage tree of C. elegans, it was actually hand drawn, and they've been able to validate it since then, of course. But like the idea was that you'd sit there and observe it, and you'd draw out like a tree based on what you're seeing under your microscope. It was pretty. It was There's no computation involved in that. So, one of the things that's interesting there is that we have data. We have like all this cell tracking data and microscopy data we can segment out, but we also have this annotation data. But we only have like this is something that was kind of done through observation. It wasn't there. Some people have done things with molecular techniques like barcoding and things like that, but there's still relationships in there that we don't know about. So there may be relationships in there that might be very interesting using this sort of graph embedding and then doing a task. And then we can actually take what we get out of that and match it up to what we know from some of the other techniques. So that might be actually interesting because we do know about the biology, and I don't expect you to know that much about the biology to start with. That's something we can build up towards because it's something that you have to sort of learn a little bit about, and then once you learn about it, you can, in past years we've had people kind of put the lineage tree, use that as like a training step. So like they'd use the data set, they'd build a classification, but then they'd also have the lineage tree to help them along. So they label the data with the lineage tree. But you don't have to do that. And in fact, it may be interesting to not do that and see what happens. And you can do that after the fact and then compare the results.
yes. Could you please share some links of mention of the text you mentioned after the meeting in Slack?
Yeah.
Yes, I want to get more understanding of that. Yes.
Oh, for like the lineage tree and things like that. Yeah, I can do that. And there are a lot of resources for it. It's just that, and they're outside of our group. But I mean, like there's a lot there though.
Yeah, and I have another question. Is about the explainability of graph network because I heard that when people gonna utilize some tools of deep learning on the application, such as some segmentation or cell tracking, they are about the explainability of graph neural network, such as why this tool can solve some task. And I'm wondering, does the team care about this question? Like if if this model can be explained by some mathematical way? Yes, because I think if we lack explainability of models, we can really trust the output of the model. But if we care much about the explainability, we cannot design some effective or efficient model structure.
Yeah.
So yeah, I'm interested in it.
Oh yeah, we are interested in that. We actually talk about it occasionally. We're interested in kind of the pure mathematics of it, I guess, or the pure machine learning of it. So we've done that in the past quite a bit. It's our main focus is on sort of the biological how you apply it to biology, but we're also interested in the mathematics of it as well, because a lot of the things we talk about here, we have to understand the math to understand how the model can be applied. So it's not not something we can just throw on to the biological data set. We have to know kind of what it's doing and optimizing, mathematically. So yeah, I think that's something that we could talk about as well, or work on.
Okay. Bradley,
Yeah.
got a question. Do we have any differentiation trees for mutant nematodes?
No. I don't know if, yeah. I don't know if it would be a matter of just taking out the cells, or if it's, because I mean, like a lot of the defined mutants, kind of what the cells are missing. Like in some mutants, they miss, they're missing certain cells in development.
Okay.
Yeah, so I don't know if it's a matter of just taking those out or what.
Is it known what's happened when you cross a wild type with a mutant?
Yeah. Oh, what happens, or?
Yeah, what happens? What do you get in progeny?
Well, you get you get the mutant, but you also get if you do a cross, say between two mutants, you get different combinations. I've done this to make a cross where you want to have a double mutant, and you can make double mutants and even triple mutants doing crossbreeding experiments. So you might,
okay.
yeah. So the wild type would be similar. You'd have that mutation in the wild type.
The reason I ask is I'm planning a paper with the editor of Biosystems on macroevolution. And in a way, we'd like to test the idea that I put forth over 20 years ago, that macroevolution is a change in the topology of the differentiation tree, and microevolution is a change which preserves the differentiation tree. So it would be interesting to see if any of the nematode data might be able to test that in some sense.
Yeah. Well, it would be, yeah. I mean, you would have, wonder if we could do that just kind of computationally. I don't know. But yeah, I mean, it seems like it would be very similar, because the only thing you're doing in a cross where you have, say, two defined mutants and you're just making one that has both defined mutations is that you're just combining them, and then when they're combined, sometimes the phenotype isn't the phenotype of both of them. It's if well, sometimes it is. Sometimes it is. The ones I did were actually a combination of both. So you'd get both sets of traits in the organism. So
okay. Yeah, I guess the starting point would be to try to work out differentiation trees for different mutants for the progeny. What do you call them? F1 and F2?
Yeah.
Okay. And see if there's any pattern there in terms of whether the progeny are fertile or not, and whether that correlates with structure of the differentiation tree or topology of the differentiation tree.
Yeah.
They do that a lot with plants. There's a whole data sets with plants. I don't know if that helps.
Huh. I don't know either. Plants get a little confusing because they're very how should I say, they're very prone to polyploid.
Right. Yeah, plants. Yeah.
And so and also I don't think anyone has worked out a differentiation tree for any plant.
Yeah.
That sounds like an interesting project.
Yes.
Yeah. So, do we have anything else? Karan, did you have something you want to present today, or?
Yeah, Yeah, I just had some more, general doubts because, as I was going through my proposal, I was thinking of things, that I can add to it. So one of the major things, after the model, like, should I add like volumetric representations of individual cells, or like, should I label each section? These kind of features that would, improve, like make it more feature rich, the final implementation of that thing, right?
Yeah.
So I was kind of looking for suggestions on these aspects. So I was thinking of maybe labeling each cell in that particular stage, as well as getting the projected surface area of that particular cell, then getting the volume based on that projected surface area. So like this was my thought process, of the kind of features that can be added to the
Right.
final implementation.
Like,
And I also had some more general doubts. Like, do all axolotl embryos have like a heavier, darker side, or is this like is it called the ventral side or the dorsal side? The darker side of those embryos.
I guess it's the ventral, right? Like, Susan?
Okay. So I think there's this big tendency, of most of them having a lot of like most of their projected like pictures are usually in the darker side. Like have like the percentage, is kind of skewed more towards the dorsal side of the embryo. So is it because it's more heavier on that side because it's a flipping microscope, right? So I think that causes it.
Dorsal ventral division is something different from the animal vegetal divide.
Okay.
Vegetal is bottom, and it's got the yolk with the high energy molecules in it that the developing embryo uses for food, basically. And then the top side is the animal section that turns into the creature, the axolotl. I don't know. Somebody else can add to this.
Yeah, I see what you mean.
Is it correct that the top side is more pigment?
Yeah, the top side usually has more pigment in it. There are
How sharp is the line between the top and the bottom?
pretty sharp. Yeah, there's a definite divide there.
Okay.
And it changes if you change gravity. If you put the animal in a centrifuge, then you'll get a change in the amount of animal section that you have.
Okay.
Yeah, it causes abnormalities in development. What do I know about dorsal ventral? Somebody asked me that actually when I was in Ricken, and I said, "Oh well, I'm just going to take pictures and look at it and find out." And he didn't like that. I'm an engineer. Like, just don't ask me these silly things. At least I know what ventral is,
Yeah.
Anyway, so there's it's a break of asymmetry in the developing animal. That's I would like to get these pictures so that maybe we can see what's going on.
Yeah.
There's been work in zebrafish for dorsal ventral development. It's in the zebrafish literature.
Yeah.
Okay.
All right.
Because I would like for the feature at least say that this particular side is dorsal low ventral. At least that was what I was thinking, for the implementation part. Yeah, obviously it doesn't make sense.
By the way, Karan, this is an interesting side point. the zebrafish is native just north of you to Burma. Okay, it's a native fish near you.
Yeah, it's this connection.
Oh,
Oh,
Oh, it wasn't my fault. I turned off my video.
yeah, it's. I'll get him back, but yeah, I guess the yeah, I think it's good to have like the annotation part. I think that's something that okay, there he is. You back?
Hello.
Hello.
Karan, we
Hello.
disappeared while I was talking to you.
Yeah.
I was saying the zebrafish, I think, is native to Burma, which is just near you.
Audio is still not.
Okay, maybe I'll use chat.
Yeah.
Could you type it? I can't hear it properly. I think
Yeah,
there's a lot
yeah.
of distortion.
Where is the chat? Oh, there it is. Oh, the chat. Okay.
Okay. So I think it's good that well, I'll type it too. Yeah, I think it's good that it's good to have annotations. And yeah, it's something we've tried to do in some of the other projects, but it's really not something easy to pull off. I think in this kind of visualization where you're navigating a sphere, you know, it's kind of like Google Earth where you have the Earth and you're zooming into different places and the maps have annotations on them, like different pieces of information. And usually the way they do that is through layers. So you can turn layers on and off as you go through. And
Okay. We reviewed a paper here about zebrafish that did the dorsal ventral development, and it was there was that
Okay.
the ball of cells that rotated, and I think it was left-right asymmetry, actually. But anyway, we reviewed some zebrafish articles. I don't have the
Okay.
I don't have them just here, but I think it was earlier. Was it November or something? Don't remember.
was it in this group or another group?
Yeah, it was in this group.
Oh,
I remember talking about it because and then I realized that I hadn't explained the whole thing, but I hadn't explained the whole thing partly because I didn't understand the paper. They hadn't laid it out well enough for me
yeah.
to say, yeah, they knew what they were talking about. Like, it was not complete, but it was intriguing anyways.
Yeah.
I can look for the paper, see if I can find it here.
Yeah. Yeah, I can probably find it too after, but I can't remember. It's like all these meetings merge, but that's yeah. See, that would be useful, I think, especially cross species, and I mean not necessarily for the proposal, but
Okay.
yeah. Okay.
Okay. Even people like modified it a lot more. Like earlier the font was different and inconsistent. I kind of narrowed down those changes, made it more easier to read. So I made those changes. Then I've added a few more points in the axis of rotation and the how approximating the angle of rotation is the portions, like getting the exact transformation formulas down there. The x and y coordinate will be changed to x, y, and z, and the x and z will have like x cos theta, y sin theta changes because we are taking within that particular axis. So I kind of added those well. So I think apart from these, the only thing that I think I could improve upon right now is just the feature description and all the annotation that you just mentioned, right? So for improving the current version of it. and any other suggestion would be like for improving the like any area that I would want to it.
Okay.
So it should have like 2-3 features, just
Yeah.
something along those lines.
Wouldn't that be part of what Hari Krishna is working on?
Yeah, I don't know, but
I mean, to place Susan's images onto a sphere requires knowing those angular coordinates. Or estimating them from mosaics.
okay. Yeah, I don't know. We'll have to read. I have to read over his proposal, so I'll see. Yeah, I'll see where we are with both proposals, and I'll give them feedback. Yeah. But you were going to mention something, Dick?
Yeah, we got a simple problem with Basilaria, and I was wondering if someone would want to tackle it. And that is, most of the movies are made at 30 frames per second now. Okay? Which is quite adequate to answer a simple question. And that is, can we quantitatively say whether the motion of Basilaria cells is smooth or jittery? Or what's called jerky.
Yeah.
So we should use. Okay? Visually, they look smooth, but your eye is very easy at deceiving you.
Yeah. Yeah.
So, this is, simple segmentation and tracking a cell versus time and plotting whether or not it's smooth.
Yeah.
And there are probably 100 movies available online to test it with.
Yeah. Yeah, there are a lot of YouTube videos of Basilaria, and we have some data on it as well. It was generated by Thomas Harpick. So
Yeah, he thinks it's smooth, but it's a visual impression, so I don't trust it.
right.
And if it is smooth, it will be different from isolated species of diatoms where the cells are isolated, because those are jerky.
Oh, right. Yeah.
Okay. So it's a small question, but it requires
Like even getting disconnected.
Yeah.
it requires tackling the segmentation problem and measuring velocities.
Yeah. All right. All right. So, did you have any other questions, Karan?
Should I go over it again?
We were just talking about we heard a lot of it, but we didn't. The last part, I think Dick asked if your proposal overlapped with Hare Krishna's in terms of projecting things onto a sphere. I guess that's what he was asking.
Okay.
Yeah.
Okay. Yeah. The main part is projecting it onto the integrated 3D model. So I kind of just focus more on that. I've just given descriptions, of various shapes it can go onto. The fair part is also text. But should I just focus on the 3D generator?
Yeah. I mean, yeah. So I think that I like you presented the weighted 3D model, I think 2 weeks ago or something, and I like that. Of course, that's a little bit different than what Hare Krishna is doing, so you find a way to balance that out, I think.
Yeah. Okay.
Yeah.
Okay. So, okay. So maybe more will work in combining both the aspects, more smoother with more smoother curves. But yeah, in the end, it depends on the final rendition of the 3D model,
Yeah.
And I'll see according to that, I'll add things.
Yeah. Okay. All right, so I'm gonna finish the meeting with a couple of things following up from last week. We talked about let's see. This is we talked about jamming phase transitions last week. So I have a whole bunch of. I'm gonna kind of quickly go through these and talk about like some things that follow up on last week. So I'm not gonna spend a lot of time on it, but for each paper, but I can send these out after. So the first one is to follow up on this idea of jamming phase transitions, which are these changes in phase that involve density of particles. And the idea that some sort of like cooling or heating process, or maybe some sort of simulated annealing. So this is a paper, nonlinear dynamics. I don't remember what journal this is, but it's jamming is just not cool anymore. And so this is kind of goes through. It's like one of these previews of different papers that are out there. So all around us, things seem to be getting jammed. We travel on a highway and we are caught in traffic jams, and it kind of goes through this. We are usually so irritated that we not really notice that a jammed state in all of these situations has common properties. So the vibrations from the pounding actually do some good in reinitiating flow. So this is where they, in this example, in factories, powdered raw materials clog the conduits that were designed to carry them smoothly. Our recourse in all these situations is to pound on our conduits and until the jam miraculously disappears. So that's the idea, that these phase transitions happen and then there's some process to unjam things, and then the jamming will reoccur again maybe, and there's this process of jamming and unjamming. Does the jammed solid then have different properties from the solids we normally encounter in the laboratory? So this is a paper from Physical Review Letters. Cates, Whitmer, Bouchard, and Clouden contend that these jam systems really belong to a new class of materials called fragile matter. These systems resemble solids because the particles are driven into a jam state by an externally applied stress. When jammed, the disordered system is caught in a small region of phase space with no possibility of escape. So this is where you have this phase diagram for jamming. This is not exclusively biological. This is for all different types of jamming phase transitions. So you have temperature, load, and density, which are your three parameters, or 1 over density, which is the inverse of density. And then you have the glass here. This is a glass state. This is a liquid state. So along this temperature and density reciprocal axis, you have glass that turns into a liquid. Along the temperature load area, you have these jammed grains turning into a load, and then loose grains, bubbles, droplets turning into jammed grains on the load reciprocal density plane. So you can see that there are all these different, you have this central region, which is the jam state. It could be glass, it could be jammed grains into a solid, and then there are all these things that are kind of, other areas that predominate. So the approach of Cates is to start from a pile of completely non-deformable particles for which strain is not obviously useful variable. Their simple model of the chain is of hard particles, of a chain of hard particles insists that the jam system cannot be considered as an elastic body. So they kind of go through this model, and they talk about because there is no obvious relation connecting stress to strain throughout the pile, Cates et al. Bypass the strain altogether and propose a relation between different components of the stress tensor. This continues to be a hotly debated assumption. So people are building models of this with particles, things like shaving foam, and they're evaluating the state and the transition. So shaving foam, for example, is jammed because the bubbles are tightly packed together under an isotropic stress, namely atmospheric pressure. If it were fragile, it would respond plastically to a shear stress, no matter how small. However, because bubbles deform, foam actually responds elastically as long as the stress is below the threshold value. Sand grains also deform slightly. Sand grains are interesting because they actually also use sand piles to demonstrate first-order phase transition, where you drop sand grains onto a pile, and then that pile at random intervals it releases stress by deforming, and you get a large avalanche of sand grains. And so this is the type of thing as the first order phase transition. In this case, they're talking about sand grains being involved in a jamming transition. So this is interesting that they're able to look at this in both ways, or you can look at that in both ways. We know from studies of liquids and glasses that a system with attractive interactions often behaves in the same way as another, that is only repulsive forces, but is confined in a container. So this is something that constrains its density. In the case of jamming, the opposite situation may be possible. That is, one might be able to replace the constraints of an external pressure or stress with an attractive interaction between the particles. Thus, a supercooled liquid can be jammed into a glass simply by lowering the temperature, not by applying a stress. When a liquid is cooled below its freezing point, its viscosity increases rapidly. Eventually, it falls out of equilibrium into a disordered solid or glass, where it only explores a small part of the phase space, just as in the case of jammed granular material or foam. So this is the phase space, and when it becomes glass, it explores a smaller part of this phase space. So this is how, these phase transitions affect the behavior of these different materials. So that's like a nice little overview. Then yeah.
Can I make a comment?
Yeah.
My graduate work was on this, but we didn't call it jamming.
Okay, yeah.
I worked on a membrane, which is an Ising lattice. And what would happen if you looked at transport across this membrane, once the concentration inside the membrane was high enough, it was an Ising lattice, the particles would attract one another and it would jam. And the transport across the membrane would go down enormously. Okay, so it was jammed in this language. But it was a phase transition inside the membrane. I did that in both two-dimensional and one-dimensional Ising lattices across the membrane.
Oh yeah.
Okay, so it's similar, and it probably fits in that category where it said if there are attractive interactions between the particles, which is what I simulated.
Yeah.
Okay.
Yeah.
Okay. So I think it fits in the same sort of thing. And it's kind of weird because from the point of view of a membrane, the jamming acts like a switch, turning the transport on or off, depending on whether the jamming's occurred.
Yeah.
Okay?
Yeah.
I can point those papers out to you if you want.
Yeah, that would be great.
That was my thesis.
Oh,
One of
yeah.
them was my thesis work. It's that old.
Yeah.
Okay?
Yeah.
We just didn't think. We've thought of it in terms of jamming in the one-dimensional case, at least, because it's analogous to what. Have you ever been at a single-lane bridge?
Yes.
Yes. Okay. They're common in New England.
Yeah.
They're often covered structures. At any rate, the traffic can only go one direction or the other, and so you end up with jamming interactions inside the bridge.
Yeah.
Okay. So that's why the one-dimensional model was interesting. Okay, so we did make that analogy.
Right.
Just never use the word jamming.
Oh yeah. Yeah, I think this is a more recent thing. Like this goes back to has a history, but I think people were kind of playing with it more now. Yeah.
I'm just saying it has precedence.
Yeah.
Okay.
So again, this paper, which is jamming phase diagrams for attractive particles, this kind of goes through. This is, I think I showed this. Well, this is the one we just saw in this paper, but I think people in last week's papers, I think at least two of them used this diagram. So I was trying to track down the source of this diagram. It's from this Nature paper back in 2001. So, this is where they kind of laid out. Actually, this is from Liu and Nagel. So this is the one that we just talked about. This is the next paper that really kind of talked about this in this using this language. So this kind of goes through talking about jamming in these type of soft active materials, and then talking about like how jammed solids can be refluidized by thermalization. So you can take something that's jammed, you can unjam it and rejam it, and there's this process.
Okay. Yeah, by the way, we can relate it to the project sort of on hold with Tom Porteous. If you look at jamming in the one-dimensional Ising membrane, okay,
Yeah.
and then you plotted that versus time, you would generate Wolfram-type patterns.
Yeah.
Okay. So it's a possible, we could possibly relate this jamming stuff to pattern development.
Yeah. Yeah, I'm just writing
You
down.
can do it in higher dimensions also, but then you have to get you have to start thinking in higher dimensions.
Right.
Like one dimension with time as the second dimension is about our limit.
Right. So that's, and then there's this, then we get into some of the things that are more biological. Actually, this one is universal scaling laws of glass, rheology. So rheology is just this study of the material. And, scale, we've talked about maybe universal scaling laws a bit in other contexts. But, with phase transition, first order phase transitions, they often use scaling laws to describe the behavior of these phase transitions. So, like I told you in before, that there's this sand pile model where you drop grains of sand on a pile and that pile displaces, with massive amounts of sand particles at random. So, but the distribution of those displacements observe the scaling law, where you get a lot of displacements of single grains after a grain has been dropped on the top, and then a very few number of large displacements. And so those can be scaled according to a scaling law, according to a power law function. So that's where scaling laws are often used. They often use them also to analyze complex networks. In this case, they're looking at glass rheology. They're looking at this universal scaling law. So the similarity in atomic molecular structure between liquids and glasses has stimulated a long-standing hypothesis that the nature of glasses may be more fluid-like rather than the apparent solid. So they're talking about this, that glass behaves like a fluid, but it appears to be a solid. And of course, it behaves like a solid if you put liquid inside of a glass. It doesn't leak out. There's like condensation on the other side sometimes, but in principle, the nature of glasses can be characterized by the dynamic response of the rheology, which is the properties of the material, and a wide rate range. But this has not been realized experimentally. Here we report the dynamic response of shear stress to the shear strain rate of metallic glasses over a time scale of 9 orders of magnitude, equivalent to hundreds of years. So they simulate this process of pressure on these glasses over hundreds of years, and then they use this type of stress relaxation experiment to do this. The dynamic response of the metallic glasses together with other glasses, in quotes, because there are different types of glass according if you study the material, that they take different forms at a molecular level, follows the universal scaling law within the framework of fluid dynamics. This provides a comprehensive validation of the conjecture on the jamming dynamic phase diagram, and that's that same phase diagram we saw before, which the dynamic behaviors of the wide variety of glasses can be unified under one rubric parameterized by the thermodynamic variables of temperature, volume, and stress in the trajectory space. So this is that same diagram that we saw. I don't know if they're going to have it in the paper. This is the scaling law here where you see the strain versus the stress. So this is the scaling, and you can see that it follows this law where the strain and stress are related. And so let's see if there's anything else in here. I don't think they actually show that diagram, but they show here it is. But it's just in a different form. So this is that same diagram. And then these are their states that they're observing. So they actually put states in there that they're observing. So that's with glass, and that doesn't have a lot to do with biology. But then we can look at embryonic tissues more broadly and consider their glassy dynamics. So this paper is by, I guess it's an international group, mostly from the US, Glassy Dynamics in Three-Dimensional Embryonic Tissues. So this paper is from 2013. And then this links a lot of this to biology and to embryos. So many biological tissues are viscoelastic, behaving as elastic solids on short time scales and fluids on long time scales. So again, we have this this transition in matter type how the biological matter behaves. So it's elastic, like an elastic solid on a short time scale, fluid on a long time scale. This collective mechanical behavior enables and helps to guide pattern formation and tissue layering. So this is something that's used in the process of, these sorts of things. Here we investigate the mechanical properties of three-dimensional tissue explants from zebrafish embryos. So these are the zebrafish embryos we've talked about. I don't know if they're using it for any specific reason. They have a lot of, they're well characterized, and they know kind of how these processes are well characterized in terms of single cells and single cell behavior. So they do this by analyzing individual cell tracks and macroscopic mechanical response. So they use a kind of cell tracking where they can track the embryo, or they can track the nucleus of the cell, and they can look at where it's moving. They can also look collectively, because in zebrafish you start to get a large number of cells that divide from single cells. So, unlike C. elegans, you have a large number of cells that aren't really labeled, but you can nevertheless track them, and you can, understand sort of how they're moving collectively. And then you can also do mechanical response experiments as well. We find that the cell dynamics inside the tissue exhibit features of supercooled fluids, including subdiffusive trajectories and signatures of caging behavior. So I don't know, I'm not familiar with caging behavior, but we develop a minimal 3-parameter mechanical model for these dynamics, which we calibrate using only information about cell tracks. So they're using just the cell track information, which doesn't have any inherent mechanical information. It just shows you where the cell is moving, and maybe the velocity. You can derive that from the cell tracks as well. This model generates predictions about the macroscopic bulk response of the tissue that are verified experimentally, providing strong validation of the model. The best fit model parameters indicate that although the tissue is fluid-like, it is close to a glass transition. And this is one of these jamming transitions into some sort of glass-like state, suggesting that small changes to single-cell parameters could generate a significant change in the viscoelastic properties of the tissue. So this is where, if you change these parameters slightly, you can change the viscoelastic properties of the tissue. I don't know if that relates to maybe in some mutants you might have this issue where the viscoelastic properties of the tissue change, or whether there are developmental perturbations that can occur that can do this. But the idea is that it's very, very close to this phase transition. These results provide a robust framework for quantifying and modeling mechanically driven pattern formation in tissues. So this is they kind of go over some of this. Let's see if we have any data here. So this is what the cells look like in our microscope. These are the cell tracks. You can see this as well. This is where they've kind of defined the nuclei and the edges of the cells. This is actually, D is two-dimensional cross-section of three-dimensional tissue simulation. So this is a Voronoi tessellation of this. So you can see that this tissue, this is the microscopy image. This is a simulation where they've taken what they call a Voronoi algorithm, which is where they find from a centroid the most optimal shape of the cell. And so you can see some of these cells have centroids like nuclei, and some of them don't. It's just a product of that Voronoi tessellation. And then they show some of these scaling. So this is two sample cell tracts extracted from experimental data illustrating caging behavior. So that's in B and C. That's here, this caging behavior, where it's along this, I guess along this path here. And then two-dimensional cross-section of the three-dimensional, that's the one I just showed. MSD data from experimental ectoderm, thin-dashed, and mesendoderm solid explants. So these are certain type of data coming from different types of tissue layers. You can see that they behave differently. And then they do some other modeling here. They're able to collect statistics of the individual cell trajectories. And then this is an image of this caging effect where you have inconfluent or neurally confluent tissues, meaning that the cells are packed together tightly and they don't have any room for like migration or slippage. So they're confluent. The cells are surrounded by a cage of neighbors, and only a high energy collective rearrangement allows them to escape the cage. So basically what that means is that your cells are packed together pretty densely, and the only way to get those cells to move around or move position. So you can see this purple cell and this green cell are in this position. The only way to get these cells to flip their position is through some sort of high energy collective rearrangement. So this means that, again, you can observe this jamming phase transition. The cells migrate into this confluent state. And then the only way to break them out of that state, to flip things around, is to have some high energy displacement. So you have to sort of displace that jamming transition, like we mentioned with simulated annealing before. You have this, like in simulated annealing, you have this equilibrium state that's reached, and then you heat up the metal again so you can pound it into a new shape. And so you have to apply energy to move the cells out of this jammed state. You can move it around. So, but this is in the biological context. So finally, yeah.
Do they relate that to this model? I forgot the name of it, for diffusion, where particles can sort of move in a local region and then make a big jump.
Oh, I think they do.
For that.
Yeah, I don't know.
But that's an old model for nonlinear diffusion.
Oh, okay. I'm not sure. I don't think it's Levy-Flayed, is it?
It might be Levy, yeah.
Yeah. Yeah, because that's, yeah. So I don't see any reference to that, but they have some nice pictures in here where they show, they show some of these physical experiments and how they do the quantification. It's kind of a nice paper from that standpoint too, but I don't think they mention the Levy-Flayed at all or anything about nonlinear diffusion.
Okay.
So
yeah, there's a tendency in some of this stuff to rediscover old work.
Oh yeah. So then that's finally this paper, the interplay between cell signaling and mechanics and developmental processes. So this one talks about cell signaling and mechanics. So it takes this idea of the jamming phase transition and puts it in sort of context of cell signaling and some of the things going on there. So again, this relates to things like positional information and other types of things that we find in pattern formation. So in positional information, it's the position of a cell that is either, sort of coordinated by within the larger context of the embryo, or that the cell has positional information as to where it should be in the embryo. So this positional information then, they talk about this in the context of biomechanics and other types of physical stuff. So this talks about the spatial and temporal regulation of gene expression and protein activity, guides cell physiology and behavior, but there's also some role for interactions with the physical and mechanical constraints on development. So they give a couple examples in here from biology. Assemblies of physical analogs consisting of elastic rubber bands and brass bars allowed embryologists to simulate gastrulation and other examples of morphogenesis in the amphibian Amphioxus, and test their ideas about cellular production of mechanical bending moments. So these are early studies that sort of tested the plausibility of physical laws to morphogenesis. So there's a rich history here of this. They also talk about they have this box of engineering principles and terms for people who are reading this. I imagine this is more developmental biologists, so they have this glossary here. And then they talk about cell shape change, cause or effect. So, one of the questions is do these changes, are they causal or is there an effect of something else? And so they kind of get into some of this like these physical forces, are they causal or are they effect of something else? The same thing goes for like the effects on cells themselves. So, this is a nice review for this sort of bringing together these themes, especially in terms of cell signaling and some of the developmental biology. So that's all I have to say about that. I think that's a good set of papers. I just wanted to follow up on some of the things we were talking about last week because I thought it was pretty interesting stuff. So Susan actually left a couple of comments here. Lisa Manning in a group has written about jamming. That I think they have like a lot of stuff on other stuff on jamming. Jamming in embryogenesis and cancer progression. That's a paper, Frontiers in Physics. And then Manning's group wrote about zebrafish left-right asymmetry, or left-right symmetry. So that's
yeah. That's the paper we were discussing earlier. It was Manning's group.
Right.
And the middle one there is a review. So
that's
a nice review.
Yeah. And so that's also cancer progression is also an interesting area for jamming, phase transitions, and physics of cells. I know that like even in like stem cells, they do a lot of studies where they look at like putting stem cells in different substrates and introducing forces, and they can actually sometimes affect the differentiation potential of the cell. So like if they put a stem cell on a certain substrate, one substrate versus another, sometimes they have to pattern it. They can get it to differentiate, or sometimes they do this with cancer cells, where they put it on the substrate and they can get it to differentiate down a certain pathway. So that might have some application to, when they're building implants and they can put them on a substrate as they're implanting them. They can guide differentiation in some way. So
that's similar. Yeah, Bradley, that's similar to. Are you familiar with the work of Beatrice Mintz?
No.
No, Beatrice Mintz would take two mice, one with cancer and one without, and she would push the cells together at an early stage and end up with one mouse.
Oh, okay, wow.
And the cancer cells then behaved and got integrated in all the tissues with no problems. Okay, but if you remove those cells and put them in an adult mouse, they would cause a cancer.
Oh, wow.
Okay.
Yeah. Did you?
I think it's MINTZ, Beatrice Mintz.
All right. Yeah, there we go.
So do you have the reference? No.
I could dig it out.
Yeah. Okay.
Yeah, it's about 40 years ago or something.
Oh, yeah.
Maybe more.
All right.
Like I said, people do the science to get paid for, and they get paid by for forgetting what happened before them.
Oh, yeah.
So,
Right.
that way you can assure there's always something to work on.
Yeah. All right. Well, let's wrap it up for today. Again, GSoC applications are due tomorrow, so be sure to submit by the deadline, submit to their portal, and I'll be looking forward to reading through and evaluating them. And then if you have any other things you want to bring up during the meeting, let me know, or bring them to the meeting next time, and we can talk about them.
Yeah, hi, Bradley.
Yeah.
Yeah, I'm wondering, is there anyone also working on the graph neural network project?
I'm not really sure. I think there may be a couple people who are going to put applications in, but no one's working on it right now. This is sort of getting off the ground. So I'm just kind of bringing in, people who are interested. So.
Okay, I see. Yeah.
Okay. Thanks for